Good morning. I'm Abraham, and I'm a PhD student from Sheffield Hallam University. I'm a third-year PhD student, and today I'll be talking about fava beans as a sustainable source of protein using a response surface methodology. My supervisors include Bipro Robert, Caroline, and Martin. And in Sheffield Hallam, I'm working with the National Center for Food Engineering and the Biomolecular Institute. So for my presentation, I'll take you through a brief introduction and some of the challenges we are facing currently with animal-based sources of proteins, and then some brief aims and objectives and materials and method, and I'll discuss some of the results I have so far on working with fava beans as an alternative source of protein. Currently, our conventional or traditional sources of proteins are usually obtained from animal-based sources such as meat, red meat, dairy sources, and fish. However, traditionally, we know that there are some sources of protein that we use to consume, which include pulses and legumes and also microalgae. But currently, our overdependence on animal bases of proteins have led to a lot of environmental and climate issues. So as I said, the current dependency on animal-based protein is expected to rise over the coming years. It's expected that by 2050, the demand for animal sources of protein will double. And why this increment? One, because of economic development and increase in population, and also as a result of urbanization. So what is the implication of the overdependence of animal bases of sources of proteins? One, in the production of animal-based proteins, usually resources such as water and land use usually take a huge amount, and also we have habitat loss as a result of soil degradation and loss of natural habitat for insect and plant and trees. How is that there's huge emission of greenhouse gases during animal productions, and in the food system, the cultivation and the production of animal-based sources account for a large amount of the greenhouse gases that are produced. So why fava beans as an alternative source of protein? One, fava beans is a very, very nutrient-dense source of protein. It contains about 30% of protein, but these proteins are easy to cultivate. It can be produced all year round. It's produced largely in the United Kingdom and all over Europe. It can be produced all year round. And one good thing about the production of fava bean seeds is that it's a good nitrogen fixation, is that it can fix nitrogen into the soil. And in the production of fava beans, like other legumes, you have less amount of land used, and also you have less amount of resources that you can use in this production. However, there are some implications when using fava beans as a seed or as a protein, just the raw seeds. As I've highlighted, in fava beans, there are some compounds known as anti-nutrients. Some of the anti-nutrients include vicin and covicin. These anti-nutrients can be dangerous to certain individuals. Certain individuals have a proclivity after taking in fava bean seeds, raw or processed. You can develop hemolytic anemia. So what can we do with these fava beans? Okay, so it's a traditional source of proteins, but then some people are very susceptible to eating it because it contains these anti-nutrients. So as part of my research is to use fava beans, extract the proteins from it, and make the proteins as a sustainable alternative to whey protein and animal proteins that can be used in different food product systems. So to produce these fava bean proteins isolate and pure proteins, such as can be used in replacement for whey protein, casein protein, and animal-based protein, we use a response surface methodology. So in the response surface methodology, and in our extraction process also, we are trying to use an ultrasound-assisted extraction method to make sure that as we are extracting the proteins from this sustainable source, we are maximizing the yield and the protein content. To do this, we use ultrasound extraction, and ultrasound is a very brain technology, easy to implement, that can aid in the extraction of protein. So the ultrasound uses ultrasonic waves. So because you are using fava bean seeds, so you mill the samples into a flour. Some of the proteins are in the cell wall, so you need to release the protein. So with the help of ultrasound, you can release as much as proteins you can. To do this, we optimize various parameters. That is the power of the ultrasound that is used, the amount of flour to water that is used, that is solid to water ratio that we use, And also the extraction duration, also we optimize that, and the total volume of the extraction container. So the main variables that we were important and focused on was the extraction yield and the protein content. We want to simultaneously get a very high extraction yield and a high protein content so that we can use this as a good replacement for whey proteins and other plant-based proteins, because usually the whey proteins are about 90% protein or 100% proteins. So we developed this response surface using a software called Design Expert software, which is available online. And then we observe the interaction between all the parameters we are using, that is the solids and the power ratio and the total volume, to see how these factors are influencing the yield and the protein content. To do this, we use a 3D interaction plot to observe all the influences of the parameters. So this is a 3D representation of the interaction between the various parameters and their influence on extraction yield and protein content. From our analysis, we realized that the extraction duration plays a significant role in getting the maximum amount of protein and as well as the extraction yield. So we observed to extract proteins from fava beans and to increase the yield, the solute to water ratio is very important. The amount of solute to the amount of water is important. The extraction duration as well, but we realized that one key parameter that was not really important is temperature. So data was not presented on temperature. So this is the result of the control method and the optimized method. So after the 3D optimization with the design expert, you can compute, and then it gives you the optimal method to use in your laboratory work to extract the highest amount of proteins. So on the left side of the graph, you would see the extraction yield and the protein content. With regards to the optimal conditions that were applied, we were able to get a protein content of 92.87%, and then the extraction yield was 19.7 compared to the control, using the same optimum conditions but without ultrasound. So without ultrasound, you can as well get to a protein content of 89% and the extraction yield of 16, but with ultrasound, you can get about 92% of protein. We also went further to investigate some of the functional properties of the proteins that we obtained. Why do we observe this functional property? Because at the end, we want to use this fava bean isolate protein as replacement in functional foods, specialized food systems such as emulsions and bakery products or in sports and stuff. So one way to find about this is to observe the water absorption capacity, which is the amount of the water it can absorb or the amount of oil it can absorb in terms of bakery system or emulsions. So with regards to the optimal condition, we observed that the water absorption and oil absorption was much higher compared to the control samples. The reason why the ultrasound optimized condition was high is obvious, because when you expose the samples, the proteins, to ultrasound, what it does is that it releases ultrasonic waves. So in a way, you can modify the structure. So plant proteins are usually global in their native, their hidden. So when you hit them with ultrasound, they tend to unfold a bit. And so in this case, you release some of the structures and then they combine with water, because usually the oxygen and OH are hidden. So when you use the ultrasound, they can come out and then they start interacting with the water and the oxygen. So in that case, could it be the reason why the optimum condition had a very high water absorption and oil absorption compared to? Yeah. To the left-hand side is the result of SDS page. So we do the SDS page to see the primary structure. That's the peptide, individual peptides that are available. So on C is the control and O is the optimized. We realized that after ultrasound treatment, there was no changes in the primary structure. So the primary structure, the carbon bonds are intact. But on the right-hand side, which is the FTR data, we can see that there is differences between the red and the green. Why? Because in the secondary structure, that's the alpha helix and the beta, the secondary structure, so primary to secondary. The secondary structure, there are changes occurring. But below in the primary structure, which is the left side, the SDS page, there's no changes. So the changes in the secondary structure was accounting for the reasons why we were having the high water absorption capacity and oil absorption capacity in the optimum ultrasound-assisted extraction method. So how can fava beans as a sustainable source of protein contribute towards sustainability? One in terms of zero hunger, it can contribute by addressing hunger by serving as an alternative source of protein, as a complementary source of protein to whey proteins and to other pulses that are available. Also, in terms of good health, fava beans is low in fat. Amino acid content is almost similar to that of whey proteins, and also it has low amount of it's environmentally friendly as well. In terms of industrial innovation, the application of the ultrasound system that we use is safe, is green, and together with the isolation process can make the fava bean as a good sustainable source of protein, very innovative, very safe, and also improve the yield of protein as well. And at the end, in terms of climate, the production, the cultivation of fava beans, like any other legumes, take small amount of land, less amount of resources is required, and less amount of waste is also generated, and we can still have our biodiversity, keep our animals, keep our forests intact as well. Thank you very much.